Hello and welcome to Insight Ophthalmology. This is Dr. Amrit welcoming you to another lecture. Today we are talking about the topography and anatomy of the retina. Okay, so the human retina basically measures about 32 millimeters from ora serrata one side to the ora serrata of the other side along the horizontal meridian. Okay, now the total surface of the retina in each eye is about 1100 millimeters square, and the average thickness of the retina, although this varies basically from studies to studies, is about 200 micrometers. Now, the retina is thickest at the posterior pole near the optic nerve head and in the macula, and as you move away from them, it gets thinner and thinner. Right? It gradually thins out at the ora serrata and at the foveola. Right? Now, if you look at this diagram, I don't want you to really mug up all these values, but remember that the retina is thickest at the optic nerve head and it is thinnest near the foveola, and the second place where it is thinner is the ora serrata. Okay? So moving on, in our video on the layers of the retina, we already talked about the 10 layers of the retina and what really forms the 10 layers and how do we actually remember them easily and logically. So the link is going to be there in the description box. You can watch that video as well. In this video, we shall be focusing on the topography of the retina, where we shall talk about what is meant by macula lutea, what is fovea, what is foveola, what is meant by the periphovea, parafoveal region, what is extra-areal retina, what is near, mid, far periphery. Specifically, we will talk about the ora serrata and also some findings which we see in the peripheral area of the retina. Right? Now, the first thing that we'll talk about is the macula lutea. Now, the macula lutea is also known as the area centralis or the central retina. It is about a 5 to 6 millimeter diameter circular zone of the retina, as which is drawn here in the diagram, and it is centered between these superior and inferior temporal arcades and the optic nerve head. Okay. Now, important point regarding the macula lutea is that it predominantly has the cones, right? And also the ganglion cell bodies are arranged in more than two or more layers within this area centralis. So that is a very important point. And more than about 50% of the total ganglion cells that are present in the retina are actually present in this area centralis. Now, the area centralis or the macula lutea, if you carefully observe, it has this term which is known as lutea. Now, the reason is why is it called so? This happens because of the presence of oxygenated carotenoids. So, lutea basically means yellowish appearance. So, macula has this yellowish color to it because of the presence of carotenoids inside of it, and these carotenoids are lutein, zeaxanthin, okay, and they impart the yellowish color to the macula. Also, they have a protective role because they will filter out all the short wavelength light, which is basically composed of the UV radiation, and therefore they also protect our eyeballs from the UV radiation. All right. Now, another important point regarding this macula lutea is that it basically contributes to about 15 degrees of the field of view. Right. Now, moving on, we have next is the fovea. Now, the fovea is about 1.5 millimeters diameter area within the area centralis, and as we all know that the size of the optic disc is also somewhat similar, and therefore we can say that the fovea is about one disc diameter in size. Okay. Now again, since the macula has only cones in it, since the macula has predominantly cones in it, the fovea region, as a matter of fact, is consisting of entirely cones. There are only cones and no rods in the foveal region, and this accounts for the high spatial visual acuity that we get over here, and is also responsible for the great color vision that we enjoy. Okay. Now, the fovea basically has a margin, a slope, and a floor to it. Okay, so it's a three-dimensional structure. As you can see in this OCT image, drawn in yellow color is the fovea. The fovea has a margin, it has these sloping edges, and it has this floor. Now, the fovea size, as I told you, is about 1 disc diameter, is equal to size of the optic disc, and it actually accounts for about 5 degrees on the visual field. So, macula is giving you 15 degrees, whereas the fovea accounts for about 5 degrees of the visual field. Now, I talked about the fovea. Now, you can see something over here. This is called the floor of the fovea. Now, the floor of the fovea is known as the foveola. Okay, so foveola is this 0.35 millimeter diameter area that you see within the fovea, and this region that I've drawn here can also be called as the floor of the fovea. Now, foveola is smaller than the foveolar vascular zone. So we'll talk about the foveolar vascular zone and what is exactly meant by foveolar vascular zone or FAZ. Now, what you should remember is that this foveola is entirely avascular and it basically relies on the underlying choroid and the choriocapillaries for its nourishment. The foveola is situated at a distance of about 4 millimeters from the center of the optic disc temporally, and it is about zero point 0.8 millimeters inferior to the center of the optic disc. So that is the location of your fovea, a very important point that is commonly asked in exams. Now, I would like you to observe this OCT image over here. So, what did I tell you? That basically this point, the floor of the fovea is formed by what is known as the foveola. Now, fovea also consists of only cones, and therefore the foveola will also consist of only cones. However, there are some histological differences over here. The cones of the foveola region are quite slender and elongated and densely packed compared to the cones of the other region of the fovea, right? So, if you can see over here, this black or hyporeflective region that you see over here. If you see in the region of foveola, it is slightly thicker compared to the rest of the retina, right? And this happens because the outer segment of the cones are quite longer and slender in this region, and that is the reason why you see thicker outer segment layer just below the foveola, right? Apart from that, if you observe carefully, the inner retinal layers, that means you can see the ganglion cell layer, the inner plexiform layer, and the inner nuclear layer. They seem to be limited just till here. Okay, they seem to be limited till this point, and they're not really present within the foveola region, right? And therefore, we can say that the inner layers of the retina actually are slightly displaced, and they basically stop at the region of foveola. And therefore, the foveola basically consists of only the outer layers of the retina, like you can see the outer nuclear layer and of course the other outer layers of the retina. The inner layers like the ganglion cell layer, the inner plexiform layer, the inner nuclear layer, they are laterally displaced in the region of foveola, right? So that is one important point that you must remember. Another important point is that it takes time for foveola to develop. It develops fully only at the age of 4 years, right? And this sloping border that I talked about here, the sloping border of the fovea, where the fovea is meeting this floor, and the floor is known as the foveola. This downward sloping border of the fovea, where it meets the floor, is known as the clivus. Okay, so again, another terminology that you must know, right? Now, when we're talking about the visual field of the foveola, the visual field that the foveola represents is about 1 degree, right? So this is a table of comparison. So macula is about 5.5 millimeters in diameter. Fovea, a region within the macula, is 1.5 millimeters, and foveola is about 0.35 millimeters. Now, macula represents about 15 degrees of the visual field. Fovea represents about 5 degrees of the visual field, and foveola represents about 1 degree of the visual field. Now let us talk about another term, and that is the umbo. Now within the foveola region there is a tiny depression in the center, and that is known as the umbo, and it is seen ophthalmoscopically as a visible light reflex, and that light reflex, shiny reflex that you see just at the center of the macula is known as the foveolar reflex, and it will be seen in every normal eye. Apart from that, what you must know is the FAZ. So what is meant by FAZ? FAZ is nothing but it is foveal avascular zone. It is a region within the fovea where the blood vessels are actually absent. Now the diameter of the FAZ would be somewhere between 250 to 600 micrometers. It could be greater as well, and it corresponds to a field area of about 1.5 degrees. Now if you would remember, fovea corresponds to about 5 degrees of visual field, whereas foveola corresponds to about 1 degree of visual field. And therefore, we can say that the limits of the foveolar vascular zone actually is somewhere between the ending of the fovea and the foveola, right? So as you can see over here, this is actually a fluorescent angiographic image. You can see this central area where the blood vessels are basically absent, this area over here, and that area where the blood vessels are actually absent is referred to as the foveal avascular zone. It's a very important landmark that you see in the angiography. Okay. Now, as you have understood what is meant by a foveal pit and what is meant by foveolar vascular zone, foveal pit is nothing but the sloping edge of the fovea where it basically meets the foveola, okay, that actually is forming a pit-like depression within the foveal region that is known as the foveal pit. Now, embryologically, what is seen is that the foveal pit and the foveal vascular zone basically they develop together, okay. And if there's an abnormality in the foveal pit, usually there will be an abnormality in the foveal vascular zone as well. Okay. Now, what did I tell you? The foveal development usually occurs, is completed by the age of 4 years. However, in some individuals, there will be incomplete development, and in those individuals, there will be absence of the foveal pit, and this condition is known as foveal hypoplasia. Okay. Now, there are some diagnostic tests which actually tell you regarding the foveal hyperplasia. So, number one is on ophthalmoscopy. You will see that the foveal region, because it is not developed properly, you will see that there is no foveal reflex. What did I tell you? The foveal reflex is basically coming from the umbo, or that extreme tip of the foveal pit. So, therefore, since the foveal pit is not developed, since there's foveal hyperplasia, there's going to be absence of the foveal reflex. Apart from that, the normal foveal pigmentation would also be absent. On fluorescent angiography, you will see that the foveolar vascular zone is absent, or the diameter of the foveolar vascular zone has basically decreased. On OCT, again, you can see that the foveal pit is basically absent, and normally what happens is that the inner layers are laterally displaced at the fovea, and it is just the outer layers which are present there. And over here, in foveal hyperplasia, that lateral displacement fails to occur, and therefore there will be persistence of the inner layers through the expected area of the fovea. Again, on the OCTA, that is your optical coherence tomography angiography, you will see that the FAZ is absent, both in the superficial capillary plexus and also in the deep capillary plexus. Now over here you can see that, do you find any shiny reflex in the foveal region? No, that is absent, right? And similarly over here, if you do an OCT scan, you can see there's no foveal pit. Normally it should be like this. And moreover, the internal layers or the inner layers of the retina are continuous in the region of the fovea. Okay, so there's no foveal pit formation as well. The next terminology that we'll talk about is the Parafovea. So what is meant by Parafovea? Now till now we discussed about area centralis or macula, and within the macula we discussed about this small circle of 1.5 millimeter diameter. Of course, this image is again not to scale, otherwise this is known as fovea, and the fovea is almost equal in size to that of the optic disc, right? And within the fovea, this blue color thing is your foveola, and within that you will have a small depression known as the umbo. Okay, now outside the fovea, if you draw another ring of about 0.5 millimeters in 0.5 millimeters thickness around the fovea, that is known as the parafoveal region. Okay, so what did I tell you? This, if this is your fovea, if this is your foveola and this is the umbo, around the fovea, if you again draw a circle of about 0.5 millimeters thickness around the fovea, that region is going to be called as the parafoveal region. Now, another important nugget regarding the parafoveal region is that in the parafoveal region, the ganglion cell layer, the inner nuclear cell layer, and the outer plexiform layers, also known as the Henle's fiber layer, is actually the thickest. Okay, so if I have to show you those layers in this image, so Henle's fiber layer is nothing but it is the outer plexiform layer. Okay, so as you can see over here, this is the outer plexiform layer. So these sloping fibers of the outer plexiform layer in the region outside the foveola is known as the Henle's layer. So you can see over here, these are the Henle layer fibers. And then above the Henle layer fibers, what do you have? You will have the inner nuclear layer, the inner plexiform layer, and then finally you will have inside the ganglion cell layer. So in the region of the parafovea, all these layers are going to be thicker. Okay, so in this region and in this region. So that is one important clinical nugget that you must remember. Now, the area of the macula which is outside that parafoveal region is ultimately called as the perifovea. Okay, so this is what has been labeled in green color, right? So again, if I have to label, this is macula, This is the ring which was demarcating your parafovea. This is the fovea. Then you have foveola. Okay, and inside that you will have umbo, right? Now the region outside the parafovea and the beginning of the macula, or the boundary of the macula, is known as the perifoveal region, right? So perifoveal region is surrounding the zone of parafovea, and it is about 1.5 millimeters in width, and therefore we can say that the umbo basically forms the center of the macula, and the periphery of the perifovea will form the margin or the outer part of the macula. So now let us try to revise. So in the retina, at the center of the retina, what do we have? We have a 5.5 millimeter zone, and that zone is known as the macula. Okay, so this green circle is the macula. Now within the macula, we have a 1.5 millimeter zone, or we can also call it as one disc diameter because it is equal to the size of disc, and this is known as the fovea. Now within the fovea, again we have a 0.35 millimeter circular zone, and that is known as the foveola. And right at the center of the foveola, we have a tiny depression which is known as the umbo. Now, don't forget that in between the foveola and the fovea, we have a zone which is known as the foveal avascular zone. All right. Now, if you draw a circle of about 0.5 millimeter in thickness on or around the fovea, now that zone is known as the parafoveal region, and the remaining part of the retina which is outside the parafoveal region is known as the perifovea, right? So what can we say? We can say that the macula basically has three zones. It has one what is known as the parafovea. The parafovea is about 0.5 millimeters thickness, and it has a perifovea around it which is about 1.5 millimeters thickness. Okay, so if you actually do the math, it actually comes to about 5.5 millimeters. Okay, so let us see how. So here you can see that this zone is our fovea, and what did I tell you? The foveal diameter is about 1.5 millimeters, right? Then around the fovea, what do you have? You have parafovea, which has thickness of about 0.5 millimeters on the all around, right? So that comes to about 1 millimeter. So add to 1.5, and then you have the perifovea, which is about 1.5 millimeter on the either side, so that will be about 3 millimeters. So if you add all of this, what you get is about 5.5 millimeter. Okay, so that was about the central retina, area centralis, or the macula. Next, we are going to talk about the area of retina which is outside the macular area. Now, this retinal area outside the macular area is known as the extra-areal periphery of the retina. And an important fact about this part of the retina is that it is rich in rods. So, when we were talking about the macula, we said that the macula area basically has cones in plenty, whereas here in the extra-areal periphery, we have rods in abundance, and also the periphery has just one single layer of the ganglion cell bodies. Okay, and the anterior end of the periphery of the retina, or we can say that the anteriormost part of the retina, is basically formed by the ora serrata. So we are going to talk about what is meant by ora serrata, but know that the distance of the ora serrata from the center of the disc on the nasal side is about 18.5 millimeters average, and on the temporal side it is about 23 to 24 millimeters from the center of the disc. All right. Now, just like we divided the central retina, we can also divide the peripheral retina. Now, we know that the macular area is about 5.5 millimeters in diameter. Now, if you draw a circle of width about 1.5 millimeter thickness all around the macular area, now that is known as your neoperiphery. Again, if you draw another circle of thickness about 3 millimeters around the near peripheral zone, that will be known as your mid periphery zone. Okay, so this is your mid periphery, this one is your near periphery. Now, beyond that, whatever you see is known as the far peripheral area of the retina. Now, this far peripheral area of the retina is about 16 millimeters from the center of the disc on the nasal side, and it is about 9 to 10 millimeters on the temporal side from the center of the disc. Okay, so I hope that is clear. So this is one way of classifying near periphery, mid periphery, and far periphery. Okay, apart from that, some textbooks actually give this term far periphery using the equator as a reference. Okay, now, if you would have seen our video on the blood supply of the eye, in that I mentioned about the vortex veins ampullae, right? Now, the ampulla of the vortex veins, they are basically situated at a distance of about 8 to 9 millimeters from the ora serrata. And remember, the outermost border of the retina is actually the ora serrata, or we can also call it as the anteriormost limit of the retina. Okay, why am I calling it as the anteriormost limit? Is because the retina or the eyeball as such we know is not a straight plane. It is actually a curved structure like this. So the retina is also curved. Here you have your cornea. So the part of retina which is going to be anterior to the equator is called as the anterior retina, and then posterior to the equator is the posterior retina, right? So over here, the part of the retina which is near the cornea, ciliary body, iris, and the lens, that is called the anterior retina, right? And this anteriormost part or the anteriormost limit of the retina near the ciliary body is known as the ora serrata. Okay. Now, coming back to the topic, so here the ampulla of the vortex veins, as you can see over here, they are about at a distance of 8 to 9 millimeters from that anterior limit or the ora serrata, right? And these ampullae are visible on indirect ophthalmoscopy. Now, if you draw an imaginary circle at the vortex veins ampullae, then what you are drawing is basically the equator. Okay. Now, some textbooks like Forrester, they basically classify the peripheral retina based on this equator. The textbook says that the retina which is posterior to this equator is known as the mid periphery or the peripheral retina, and the retina which is anterior to the equator will be known as the far retina, okay, or the far periphery. Okay, now there's another way of talking about the anatomical equator, and that is using the limbus as a reference. So, the anatomical limbus is situated at a distance of about 14 to 15 millimeters from the limbus. Okay, another high yield point that you can remember. Now, this was the video I was talking about, the blood supply of the eye, and the link is going to be there in the description box. So now we are going to talk about the ora serrata in detail. Okay. So as I explained to you, the anterior limit of the retinal tissue is formed by the ora serrata. Now we know anterior to the retina or anterior to the ora serrata, what is present? It is the ciliary body. Now the ciliary body has two parts, an anterior part which has the processes coming out of it, which is known as the pars plicator, and the posterior smooth part of the ciliary body, which is known as the pars plana. Now at this point, if you're not very confident about the anatomy of ciliary body, we do have a video on that as well. Okay, so the ora serrata basically forms that boundary between the pars plana and the neurosensory retina. At the ora serrata, you can say that the neurosensory retina basically becomes continuous with the columnar non-pigmented epithelium of the pars plana. Now, if you can see this diagram over here, the imaginary circle drawn at the ampulla of the vortex veins basically is the equator. And then you can see drawn in orange color is your ora serrata. Now, if you would carefully observe, the ora serrata actually is situated at a distance of about 18 to 19 millimeters on the nasal side from the optic disc, whereas on the temporal side, the ora serrata is situated at a distance of about 23 to 24 millimeters. Now, again, if you carefully observe, the ora serrata actually has this more scalloping edge on the nasal side compared to the temporal side. So we can say the ora serrata is more serrated on the nasal side compared to that of the temporal side. Alright, so it has a scalloped appearance, it is more serrated nasally, and also something else also happens nasally, that the ora serrata is situated much more anteriorly compared nasally compared to the temporal part. So distance from the limbus of the ora serrata on the nasal side is going to be about 6 millimeters, and distance from the limbus on the temporal side is about 7 millimeters. Okay, so this proves that the ora serrata is situated more anteriorly on the nasal side of the eyeball. All right. Now, why do we see that serrated margins of the ora serrata? Why does it have that scalloped appearance? Okay, and that happens because of these projections. So over here, this is the retina, and this is your iris. This is as if you're looking from inside, as if you have cut open an eyeball and you're standing within the eyeball and you're looking straight from inside. So this is your pupil, this is the iris, and this part here with the ridges is your pars plicator, and this brown color things is the pars plana. And here these whitish processes you can see, right? So these are the processes which are coming from your ora serrata. This part is the ora serrata, right? So ora serrata, as it merges with the pars plana, it actually sends these tooth-like projections into the pars plana, and these are known as the dentate processes. Okay, you can see over here, these are the pointed ones are the dentate processes. Okay, and these rounded tips of the pars plana are known as the oral base. Okay, so these hemicircular things are known as the oral base. So I hope that is clear what is meant by dentate process and the oral base. Okay, now let us see a few diagrams and try to understand. So this is your retina and this part will be your pars plana. Okay, and again you can see that the retina is actually the ora serrata region of the retina is projecting these tooth-like processes which are known as the dentate processes. Okay, so these tooth-like processes will be dentate processes, and in between the dentate processes, the part of the the part of the pars plana is known as the oral base. Right, again on an ultra wide field imaging, again you can see these ones here marked by the arrow are the dentate processes, and in between that what you see is the oral base. Okay, now marked with asterisks here is a meridional fold, so we're going to talk about it again in a while. Now sometimes what happens is that the dentate processes will actually move like this, and they are going to meet the other dentate process, the adjacent dentate process, and in this process and in this meeting up with the adjacent process, what they do is that they will enclose a part of the pars plana within itself, and that is known as an enclosed oral bay. So it's an oral bay which has been enclosed by two adjacent dentate processes, right? So there's an island of pars plana enclosed within the retinal tissue. So you know that this dentate process is the retinal tissue which is coming from the ora serrata, right? Now, if you can see this diagram over here, this picture over here from retina, this is actually from retina image bank. Okay, so here you can see this part, this is the retinal tissue. Now, sorry, this is the pars plana tissue, and the pars plana has actually been, a part of it has been enclosed by the retina. So this is the retinal tissue, this is pars plana tissue, and over here is the pars plana again. So importance of enclosed oral bay is that they might mimic a retinal hole. Okay, so that is one thing that you should remember. Again over here, one more image showing that these dentate processes are actually enlarged and they are basically enclosing an oral base. This is again an example of enclosed oral bay. So if you look carefully, you can see these drusen-like structures. Okay, so these drusen-like structures over here, okay, they are known as the oral pearls or the pars plana pearls, right? So these drusen-like structure basically appear at the junction of the retina and the ciliary epithelium, and mostly they are along the dentate process, and histologically they are basically formed under the RPE and near the Bruch's membrane. Okay, that's the reason why I'm calling them drusen-like, right? So these oral pearls are actually present in about 20% of the eyes. They don't have any sex or age predilection, right? So again, another image which is showing these small pearls. You can call them pars plana pearls or you can call them oral pearls as well. Another finding that sometimes you might find is known as the meridional fold, right? So sometimes you will see these full thickness radially oriented folds of the retina. So normally the retina is like this flat, but sometimes it might get folded on itself and gets thickened like this. Now these are, when this happens, it forms a fold, and this fold is called meridional fold. This meridional fold is actually oriented 90 degrees to the ora serrata. So you can see over here, this is a meridional fold in the diagrammatic representation, and this is again a meridional fold. Now this thickened retinal tissue actually extends into the pars plana region, right? Now this is also present in about 26% of the population, and they can actually be bilaterally present as well. And the most common quadrant where you find these meridional folds is in the superior nasal quadrant. Now this is an ultra wide field image. Okay, so here also you can see that these thickening are the meridional folds, and on high magnification again you can see these are the meridional folds. Now these meridional folds actually have not been significantly associated with any peripheral retinal pathology, although sometimes it has been suggested that you might find some small retinal holes near the meridional folds because of the termination of the vitreous over there and because of some of the vitreoretinal traction component, right? Now, before we go to other findings, let me tell you one more important clinical point, and that is why do you see most of the degenerations at the ora serrata region, right? Now, that happens because the ora serrata region actually acts as a watershed zone between the anterior and the posterior circulation. Okay, and therefore this region actually has just a single layer of the blood vessel, and moreover the thickness of the region is also very less, and that is the reason why the degenerations are more common within the ora serrata. So we talked about the meridional fold, and now let's talk about what is meant by meridional complex. Okay, so complex means that there will be multiple components. Okay, a meridional complex is formed basically by an enlarged dentate process, as you can see over here. And if this dentate process aligns with the ciliary process, which is here like this, and it aligns with the meridional fold like this, so all these three structures together will form what is known as a meridional fold. Okay, so a meridional fold is formed by an abnormal alignment between the dentate process, which is usually enlarged, and that dentate process is continuous with an enlarged ciliary process and a meridional fold, right? Now whenever they are present, it also leads to actually retinal excavation in the corresponding meridian. So this meridian will have retinal excavation. Okay, and these meridional complexes are found in about 16% of the population, and they can be about they can also be seen bilaterally, right? Again, the most common site where you find them is in the superior nasal quadrant. Another structure that another findings that you can see in the periphery is the presence of a cyst in the pars plana region. Now they are quite different from the pearls that we saw. Okay, now these cysts can be small cysts or they can be really large cysts. They are basically these bullous elevations that you can see over here, and they're looking clear because they're filled up with the hyaluronic acid, right? And these are formed by the separation between the non-pigmented and the pigmented ciliary epithelium. Okay, so that is one thing that you must remember. This picture is actually from retina bank, and here you can see the sparse planar cyst, and this cysts were actually found during the vitrectomy surgery for rhegmatogenous retinal detachment. Okay, so there has been a peripheral indentation that has been applied. So you can see over here, these are your ciliary processes, and this is the pars plana region, then again the ora serrata, and then you have the retina. So in the pars plana region you can see this cyst. Okay, now, many times you might actually be confused. Sometimes people get confused between retinal detachment, retinoschisis, and the pars plana cyst. And a very easy way to differentiate them is by observing the blood vessels. So, normally in a retinal detachment, the blood vessels are going to be, they're going to follow the contour of your detachment. They're going to be moving with the detachment. Whereas here, the blood vessels will be found underneath this cyst. Okay, so if you trace this is how the blood vessel is, and here also the blood vessel is like this. Okay, I know the picture is not very clear, but in a retinal detachment you might not be able to find the blood vessels below the detachment, whereas in case of pars plana cyst you will be easily able to track the pathway of the blood vessels, right? And how can we not discuss about the nerves and the veins that are seen in the peripheral retina? So this is seen usually in the wide field imaging. So you can see over here, these are the ampullae of the vortex veins, and this horizontal line that you see here is basically the long ciliary nerves, And these ones, the radially oriented ones, are the short posterior ciliary nerves, right? Again, here you can see this one is the long posterior ciliary nerve. Now, sometimes what happens is that there might be, in some cases, a normal variation of the RPE distribution. That means the RPE might be densely present near the central retina and the mid peripheral retina, and in the far periphery, the RPE, the pigment might not be distributed as densely, right? So this leads to this variation in the color of the retina, and that might actually might be falsely diagnosed as retinal detachment or retinoschisis, right? So that is one point that I want to tell you about the variable RPE distribution. Okay, so that was regarding the topography of the peripheral retina and central retina as well. I hope you found it useful, and if you did, kindly like and hit that subscribe button. That really makes a difference for the channel. And that's all for today. Thank you and have a nice day.